the first thing that I want to do today is spend some time going over this question, right? So the question is, right, explain how metabolic pathways are regulated. Somebody answer that for me. How do we regulate metabolic pathways? Through the use of what? Don't all jump in. Cole. How do you regulate metabolic pathways?
You don't know how to pronounce the enzymes like allosteric.
Yes, perfect. All right. So metabolic pathways are going to be regulated through allosteric enzymes, right? And we call this allosteric regulation. And one of the things that we are about to see right now is that when the end product of the metabolic pathway builds up, that is going to shut off the metabolic pathway. Does anybody remember what that process is called when the end product inhibits the pathway?
Feedback inhibition.
Feedback inhibition. All right. So what I thought I would do right now is make a little drawing that shows how allosteric enzymes work and how feedback inhibition works in order to regulate metabolic pathways. Remember, regulation is extremely important. Would you want all your metabolic pathways running all the time? No. It would be complete chaos within your body, right? And you would not be effectively managing your energy and managing your resources if all your metabolic pathways were going all the time. So we need to have the ability to control metabolism. We need to have the ability to turn pathways on, to turn pathways off, depending upon whether or not we need them running. Does that make sense to all of you? So this is arguably one of the most important lessons in this entire chapter, is how are our metabolic pathways regulated? Right? And they are regulated through allosteric enzymes. So a pathway might look like this. You go from A to B to C to D, right? This is a metabolic pathway. How many chemical reactions are in this pathway? 3 chemical reactions. 3 chemical reactions. 1 chemical reaction here, 1 chemical reaction there, 1 chemical reaction there, right? So you have 3 chemical reactions composing this pathway. So how many enzymes are required? 3 enzymes, right? 3 reactions, 3 enzymes. So you have enzyme number 1, enzyme number 2, enzyme number 3. We're all following so far? Now let's say that this pathway is anabolic. If this pathway is anabolic, is it consuming or releasing energy? It's consuming. And if we look at these reactions, are these reactions endergonic or exergonic? They're endergonic. They're consuming, right? And the goal of this pathway then is to build what? What are you building? What's the end product? The end product is D. Right? This is the starting substrate, right? This is what's going into the pathway, right? This is your product. D is the goal of this metabolic pathway. Remember, we described this pathway as being anabolic, right? Anabolic means it consumes energy to build something, right? And in this case, D is what is being built. Make sense? So energy is consumed. You're investing energy more and more and more, and you are building more and more D. That is the goal of this metabolic pathway, to construct and to build D starting with A. And this pathway requires 3 different chemical reactions to do that. Are we okay? Now, if we wanted to turn on this pathway or to turn off this pathway, we do that by turning on or off an allosteric enzyme. Now, in this case, which one of our 3 enzymes would be the allosteric enzyme? Enzyme number 1. Right? Enzyme number 1 is going to be our allosteric enzyme. It is the enzyme that has the ability to be turned on or turned off. So an allosteric enzyme might look something like this. So this right here is your active site. That is the site that the substrate is going to bind to. This right here is your allosteric site. Meaning that is the site that's going to be used to turn the enzyme on, or to turn the enzyme off. So this allosteric enzyme is going to be controlled using either an activator or an inhibitor. So an activator is going to bind to the allosteric site, and it's going to lock the active site in its active form, meaning that enzyme is turned on and it can catalyze the reaction. Does that make sense to all of you? An inhibitor is going to do the opposite. An inhibitor is going to bind to the allosteric site, and it is going to turn off the active site. Right? So the inhibitor is going to bind to the allosteric site, and it's going to essentially turn off the active site of the enzyme. So the enzyme can no longer work, can no longer catalyze the reaction. We're all doing okay? Now notice, I did something really clever. What did I draw as my inhibitor? D. Did we all see that? Yeah, brilliant, right? Now I made the inhibitor D. Because in this pathway, when D builds up, D works as the inhibitor, which is going to shut off our allosteric enzyme. And that should make a lot of sense to you. Because if we have enough D, do we want to waste energy making more? No. So you want to turn off this pathway until you need to make more D. Doesn't that make a lot of sense to you? So when buildup of the end product shuts down the metabolic pathway, what do we call this entire process? Go ahead, Nick, you said it before.
Feedback inhibition.
Feedback inhibition. Right? Which is an extremely important term for you to know. Right? Feedback inhibition is how we regulate our metabolic pathways. When the end product builds up, the buildup of the end product is going to shut down the entire metabolic pathway. When you shut down a pathway, you want to shut it down from the beginning. So that means our very first enzyme is going to be an allosteric enzyme, which has the ability to be turned off. Right? So D works as an allosteric inhibitor, shutting off this allosteric enzyme, turning off this metabolic pathway. When will this metabolic pathway turn back on? When there's no more D. Because when you have no more D, that means you have no more what? You have no more inhibitor. Good. So once you run out of inhibitor, then the enzyme goes back into its active form, and this pathway turns back on, and you can continue to produce more D. Now, what do you think? I think that makes a lot of sense, right? So feedback inhibition and allosteric enzymes are used to regulate all of your metabolic pathways. Are we all very clear with this? Right? Really important for you to know and for you to understand. Good. Now there's only one or two small things left to cover in this PowerPoint. Let's open it up. Now one is what Aaron mentioned before, is something called cooperativity. So cooperativity is doing the opposite. Cooperativity is not regulating. Cooperativity, hey, you throw it out, is instead amplifying the response of an enzyme. So in a way, it's almost like positive feedback. Does that make sense to you? Now, earlier, right, last week, I showed you this image of an allosteric enzyme. And when we looked at that allosteric enzyme, how many active sites did we say it contains? 4 active sites. Do we all see that? So when one substrate binds to the active site, how many more substrate can then bind to the active site? 3 more. Do we all see that? So every one substrate binding will then cause 3 more, which is just going to make that response occur faster and faster and faster and faster. Does that make sense to all of you? We call that amplification cooperativity, right? It's going to make the response of the enzyme occur faster and faster. So for every one substrate that binds, that's going to allow 3 more substrate to bind, amplifying the response of that enzyme. All right. 2 little things we have to add, and then we are done. Number 1, evolution. Every part of this course has an evolution component of it. An enzyme is what type of macromolecule? Protein. What determines the function of a protein? Its structure. What determines the structure or the shape of a protein? Its sequence of amino acids. Take it a step more, which we discussed. What determines the sequence of amino acids? The sequence of what, Alex? The DNA, right? So if your DNA changes, we call that a mutation, right? If you change the DNA, that changes the sequence of what? Amino acids, which then changes the what of the protein? Its structure, which then changes the what of the protein? Its function. So always remember that sequence. If you change the DNA, you change the sequence of amino acids, you change the shape of the protein, you change its properties. You change what it's able to do. Now think about that in terms of an enzyme. Most likely, if a mutation occurs in the gene that codes for an enzyme, what is that most likely going to do to the enzyme? Do what, Aaron? Make it stop working, right? Most mutations are what? Are bad. Most mutations are bad, right? Chances are, if you have a random mutation that changes the shape of an enzyme, it's going to render that enzyme non-functional, useless. And the thing that I think of right off the top of my head is people who are lactose intolerant. Do you have any lactose intolerant people in here?
I had a cat that was lactose intolerant.
Okay, so we have a lactose intolerant cat. If you are lactose intolerant, you cannot produce an enzyme called lactase. Lactase is the enzyme that breaks down lactose, right? Does that make sense to all of you? So if you are lactose intolerant, that means you have a mutation in the gene that codes for the lactase enzyme. And that mutation renders that enzyme non-functional. As a result, you can't break lactose down when you consume it, and it gives you diarrhea because you can't digest it. Make sense to all of you? So in most cases, mutations are bad, and they make enzymes non-functional. But what if a mutation was good? What if that mutation altered the active site of an enzyme that allowed it to do something new and beneficial, right? That mutation might be passed down. And that mutation might give that organism the ability to do something that it wasn't previously able to do, right? Like maybe it can digest something now that it wasn't able to previously digest, or some new novel thing that was beneficial. You can see how the evolution of new enzymes can come about over time that allow organisms to now do new things and catalyze new reactions that might be beneficial to them. Does that make sense to all of you? Now let's take this back to a cell. We spent some time learning about all of these different organelles that are inside of cells. Organelles like mitochondria, chloroplast, smooth and rough endoplasmic reticulum, Golgi apparatus, right? And we said all these organelles are basically made up of phospholipids, nothing fancy. What is the purpose of a cell organelle? Remember how I described the body to you. The body is a giant vat of what? I've used this phrase a number of times. The body is a giant vat of what? Chemical reactions. Chemical reactions. The cell is a little vat of chemical reactions, right? So what do you think the purpose is of having cell organelles? What do cell organelles do? They localize chemical reactions. Organelles are ways of having different chemical reactions take place in different parts of the cell, right? So my question to you is this. How do you localize chemical reactions? How do you make sure that certain chemical reactions only happen in certain locations? What do? You localize their what? Close. Remember, substrates are going to probably come into or out of the cell through diffusion and facilitated diffusion and all that stuff. You localize their enzymes. When you think about what cell organelles are, they're just little structures made up of phospholipid membranes, right? So how do different organelles do different things? Different organelles hold different enzymes. And by holding different enzymes, you perform different chemical reactions. We're going to learn all about the mitochondria next. And the mitochondria is the site of cell respiration. The reason why the mitochondria perform cell respiration is because it holds all the enzymes that perform cell respiration. The reason why the rough ER modifies proteins is that it holds all the enzymes that modify proteins. The reason why the smooth ER detoxifies is that it holds all the detoxifying enzymes. So when we think about a cell, when we think about all these organelles that we learned about, organelles are just ways of localizing chemical reactions. You localize chemical reactions by localizing enzymes. Do we all feel pretty good? And this is the end of the video for today.